Hi there, welcome to Honors Biology Unit 1. My name is Mr. Gappy. This is Intro to Bio and Organic Molecules. This is the first lecture of this chapter. Let's begin. Well, the chapter begins by talking about scientific method and the scientific process. You may have heard it both ways, all right? I have a really good picture on the next slide. I'm going to explain all of these terms on the next slide, basically. They go a little bit better with a picture. All right, here we go. Scientific method begins with something that is observed. You're using your senses. In this case, the senses were the eyes. This person was looking down and they notice, there's some seeds, there's plants that are growing, there's some soil. All right? So they're observing, they're seeing stuff going on. Based on this observation, this person says, I got a question. All right? So these observations are leading them to ask a question to themselves. What type of fertilizer would work the best? Okay, which fertilizer would work the best? So that's their question. This question then needs to be answered. And when is it answered? It's answered as a hypothesis. So if the question is which type of fertilizer works the best, the hypothesis would be, in this case, they'll say, oh, well, fertilizer A works the best. That answers the question. Well, a hypothesis leads to what's called a prediction. The prediction is pretty much saying, well, if your hypothesis is true, what would happen for the most part? So in this case, which type of fertilizer works best? Well, let's go straight to the prediction in this case. Well, if fertilizer A works the best, that's the hypothesis. Fertilizer A works the best. If fertilizer A works the best, then plants grown with fertilizer A will grow the fastest. So that's what's going to happen as a result of the hypothesis. Then that leads to the experiment. So they set up the experiment and they have fertilizer A, they're comparing it to fertilizer B, they're comparing it to fertilizer C, they're looking at the plants, they're looking at how they grew. This is the experimental data they have. And from this data, they can get a conclusion. Well, what is a conclusion? Conclusion wants to know, well, based on your experiment and your design of your experiment, did you support that original hypothesis that fertilizer A works the best? Or did you support that prediction that you made earlier? And in our case here, well, look, fertilizer A, oh, look, it did grow the best. They did grow the best. And how do we know they grew the best? Well, because they grew the fastest. And that was the prediction that was made. And so in this case, the hypothesis was supported. When a hypothesis is rejected, it's not a bad thing. Sometimes when a hypothesis is rejected, science might develop things like different medicines and different drugs are made that way. A lot of the medicines on the market today came about by accident. A drug was being made for one ailment and they realized it was helping people with another ailment. So you may have rejected your initial hypothesis that your drug would help a certain group of people, but you found out that it helped a different group of people by accident. So rejecting it isn't necessarily a bad thing. It can help you reformulate a hypothesis and test it again and see what direction it can lead you. Now, when you keep getting the same conclusion or you keep supporting the same hypothesis, and many scientists are also doing the same thing over and over again, this keeps taking place all over the world, well, then it becomes a theory at that point. So a hypothesis is how it begins, okay? And then if you keep getting the same conclusions, eventually you have a theory. And it has to be supported by many, many, many people all over the planet. So what is biology? Because this is your biology class at Valhalla High School. Well, what is it? Well, biology is the study of living things. All right. And what are the living things? We are not going to study all the living things in great detail due to time constraints, but we're going to study bits and pieces of all the living things. What are we going to learn about? Well, we're going to learn a little bit about bacteria. All right. And there's two groupings of bacteria, and they're part of this grouping they're called the prokaryotes. You have eubacteria and archaebacteria. These are the kind of bacteria you'll recognize as the kind that make you sick. All right, the germs, the E. coli, the salmonella, the kind that get you sick. They live in really harsh and extreme environments, like the bottom of a hydrothermal vent at the bottom of the ocean, very salty environments. These guys can live in very tough, acidic environments. That's archaebacteria. Okay, the two groupings of prokaryotes. All the rest of them are what's called eukaryotes. They're more complex cells. We are part of that grouping. Animals, plants are in that grouping. Fungus are in that grouping. Okay, and protists. Protista, this is a kind of a very broad group. They're very different from each other. I kind of joke around and always say, usually when we discover a eukaryote and scientists don't know where to classify it, they kind of put it in protista. In case you're wondering, algae, which are kind of like plants of the ocean, the photosynthesizers of the ocean, well, they're protists. They're not plants, but they do photosynthesis and they're green in a lot of cases. So there's protists that can do photosynthesis also. There's some bacteria that can do photosynthesis also. So how do you organize life? Well, life can be organized from, in this case, small all the way to really big. All right. In this case, really big would be the life sphere of the planet, the area of the planet that supports life. And then that gets broken into smaller systems of, well, smaller sections of Earth would be ecosystems. All right. Within ecosystems, you have communities, just living things interacting with each other. All right. So you have non-living and living, non-living and living, non-living and living, living within living. Population is one species. That one species becomes an organism. Okay. An organism is broken into many different systems. All right. Those systems make up organs. Organs are made out of tissue. Tissue is made out of cells. Cells are made out of, these little parts are called organelles, and organelles are made out of molecules, and molecules are made out of atoms. Even atoms can be broken down smaller, but this is how life is organized. In this case, I went from big to small. Now, what are the things that make up life? Well, there's general scientists and biologists have come up with a general consensus on what and how they define life. In order for something to be considered alive, all right, it needs to follow these general guidelines. And I'm not going to lie, there's sometimes there's rules that can be broken by certain living things. All right, there's always exceptions to the rule when you're talking about the characteristics of life, but they're generally smaller exceptions. So let's talk about them. Scientists, the first consensus is they believe that living things as we know them, as we know living things, they're made of cells. They might be simple cells, prokaryotic cells. They may be more complex cells, eukaryotic cells, but they're made of cells. Living things, they respond to things. And the thing that causes the response is the stimulus, right? So they respond to things in their environment. And those things I said are the stimuli. Well, the reason they respond is they're trying to maintain a balance in their life and in their internal conditions. If they don't have balance within their internal conditions, they can't do the things they need to do to stay alive. And maintaining that balance is called homeostasis. Metabolism is a fancy word. You guys usually think of it as how quickly you burn through your food, but it's bigger than just burning through your food. You're burning through your food so that your body can do all the chemical processes that it needs to do. You are a giant bag of chemical reactions, and all those reactions combined are your metabolism. A major part of your metabolism, yes, is making energy. All right. All living things we know of, they grow, they develop, they can reproduce and continue themselves, and they change through time. This has to do with adapting and evolution, basically. So we'll talk about that in a later chapter. So living things we know of can do this. Scientists have tried to reproduce life in a laboratory, and they've come up with things that look like bacteria, but they generally cannot do many of these different characteristics of life. Today, to end today's lecture video, we're going to talk about experimentation. So this is a major part of the scientific method, is how to design an experiment that is viable, that is fair, that is logical. So it begins by controlling the experiment. That means you need multiple groups. You can't just say, "Oh, I'm gonna test something. I'm gonna grow plants on a fertilizer, and now I know that fertilizer works great." You need to compare it to something. So that's what you're doing when you're creating a controlled experiment. It begins by setting up groups. You're going to have control groups and experimental groups, and they have purposes. The control group main purpose: comparison, comparison, comparison. You need a group that we call normal, and that you can compare your other groups to. Without this group, there is no logic behind what you're going to do. The experimental group or groups, you can have more than one, are the groups that are getting tested, basically. You're actually doing the experiment on these groups. Now, variables, you've heard the term variables. I know a lot of you come to me, and variable, when you learned it as a child or in earlier grades, was always the thing that you were testing in your experiment, was the variable. Well, that's kind of not a great definition. A variable is anything that can affect an experiment, basically anything. So a variable, by definition, isn't only what you're testing, it's also stuff you're not testing. Okay, so you learned it as what you're testing. Well, there's different kinds of variables. Some are the kind you're testing, called independent variables or experimental variables. Some the kind you're not testing, called control variables. And I'm going to explain them to you in better detail in an example in a second. All right. The dependent variable. This is what you're measuring in your experiment. So the independent variable is what you're testing. That test leads to measurements in your experiment. So what happened? What were the results? That's your dependent variable. Right? And we're going to explain all that in 1 second. I'm just showing you a picture that says, all right, start with 500 patients. Well, how do you set up an experiment? Well, first off, you divide them in half. Let's put 250 in one group, 250 in another group. Well, they get the drug. We're testing a medicine. They don't get the drug. Well, they'll get a placebo. They think they're getting the drug. They'll get a sugar pill, for example. And after 24 weeks, they look at what happened. Did these patients, did they get better? Did they not get better? And then they statistically analyze it to see if your experiment was valid. So let's look at an example. All right. Suppose, and by the way, you are going to get questions like this on your test. I'm not going to ask you generally what the definitions are of those things. I'm going to give you an example, and I'm going to ask you in that example, what is the independent variable? What are the control variables? What is the dependent variable? What is the question that you're trying to answer? Okay. So here we go. Suppose you're asked to design an experiment that tests the effects of a new protein supplement for weight gain in dogs. All right. So you set up, in this case, two groups: control group, experimental group. All right. So we're going to have German Shepherd puppies in the control, German Shepherd puppies in the experimental group. We cannot have different kinds of puppies here. If we had different kinds of dogs like German Shepherds and Poodles, we are now adding an additional variable that we are testing. That is not a good experiment. A good experiment tests one variable at a time. So German Shepherd puppies are something that can affect the experiment. So therefore, we do not want to affect the experiment. This experiment is about the effects of protein. Okay, this experiment is not about the effects of what type of dog. All right, so German Shepherd puppies, German Shepherd puppies. Control group, they sleep 12 hours a day. Experimental group, they sleep 12 hours a day. Once again, we need to keep these variables controlled. Right, that means we need to keep them the same. They eat 3 pounds of beef, they get 3 pounds of beef. They walk for 45 minutes, they walk for 45 minutes. They live indoors, they live indoors. The control group does not get what we are testing. What we are testing is the protein. They do not receive protein. That's the control group. The experimental group, they do get the protein. Okay, this thing that they're getting, the thing that we're testing, this is the independent variable. So what is the independent variable? That's the protein right there. What are the control variables? Well, these are the things that we keep controlled in the experiment. So there are things that can affect the outcome that we kept the same. German Shepherd puppies, German Shepherd puppies. Control variable number 1. Sleeps 12 hours a day, sleep 12 hours a day. Control variable number 2. Eat 3 pounds of beef, eat 3 pounds of beef. Control variable number 3. Walk 45 minutes, walk 45 minutes. Control variable number 4. Live indoors, live indoors. Control variable number 5 in this case. So these are control variables, these are control variables. Both groups receive control variables. The experimental group also then receives the experimental variable. In this case, the protein supplement, which is the experimental or the independent variable. So in this experiment, what would you measure if you were the scientist? Well, the dependent variable is what you would be measuring, and you would be measuring the weight of the dogs. All right? We're going to stop there today.